After we obtain the data from the FE matrix, we have to do the quality checks also. And in case of FE matrix data, the quality control and other processes of quality checks are performed on the raw data before normalization. And these quality checks includes the RNA degradation, expression for control genes, scaling factors, percentage of present genes, and average backgrounds. So these quality checks must be performed before proceeding for the normalization. And for this purpose, two different types of tools are used. One is the FE and the other is the simple FE tools. Apart from this, the box plot, hierarchical clustering, and non-metric multi-dimensional scaling can also be added in the quality control checks that can enhance the quality of the process. And for this purpose, dat.m command is used, that is present in the Lima package softwares. For the quality control of the FE matrix data, first we have to check the RNA quality. And for this purpose, we have to see whether the RNA was degraded when the FE matrix experiment was performed. For this purpose, a command FeRNA deg means our FeRNA degradation, and afterwards brackets. In the bracket we have to add the data source, and this command is used in the tool known as the FE tool. And the other descriptives can also be carried out by using the command QC and then brackets. And these commands are present in the other tools that is known as the simple FE tool. And both of these tools, the data when we use is imported by the help of the FE batch object. So we have to open the library FE, and then we have to use the tool simple FE, and then we will acquire QC data, and then we will see whether the RNA was degraded or not by using this command. And it might be interesting to see that what kind of information objects DEG and AQC contain, but typically we want to examine a visualization of the result. Plotting the general quality control statistics is simple. Then we use the command plot AGC, and we will get the quality control diagram, and we will see here. This is the QC scan plot reports quality control parameters for the chip. So we can see here different chips are present, and they are separated by vertical gray lines. This is vertical gray lines. The red numbers, we can see red numbers. On the left report the number of the probe sets with the present flag and the average background on the chip. The blue region, this is blue region. In the middle denotes the area where scaling factors are less than threefold, because we always consider the significant data that is less than threefold or higher than threefold. Either it is down-regulated more than threefold or down-regulated more than threefold. So bars that ends with the point, we can see here small bars, and they end with the point denotes scaling factor for the chip. And the triangles, we can see here small triangles, denotes the beta actin 3' to 5' ratio. And the open circles, these are the open circles, they are the gap DH 3' to 5' ratios, and we know that these are the two housekeeping genes, beta actin and the gap DH. They are housekeeping genes, and they are used as a control because these genes have the constant expression values. Either we use the control or we use the treated samples. So these are used to maintain and to see whether the number of the and the amount of the RNA loaded, they should be same in both control and the treated samples. So the deviant chips are therefore easy to pick off by their red coloring. So here all scaling factors are within the acceptable range, but some of the housekeeping genes, as we told, is GAPDH and the actin, fall outside the acceptable range. So we can see this is fall outside the acceptable range. However, most of the chips having very deviant control genes expressions are samples from cancerous tissues, and the quality control genes might represent the large gene expression changes typically displayed by the cancer tissues. So this is a very good control, and we can say that these chips can be used for the further processing.